Can one radiopharmaceutical realistically go after 5 different cancers at once? That sounds like science fiction, but it's happening. Meet NeoBOMB1, a new theranostic that uses Gallium-68 PET to find tumors and lutetium-177 therapy to destroy them. Today we're diving into science, the clinical trials, and why this could mark the beginning of a whole new chapter of nuclear medicine.
GRPR and NeoBOMB. 1. What are we targeting? GRPR-101. The gastrin-releasing peptide receptor is overexpressed in several tumors, especially subsets of prostate and breast, and is also reported in lung, GIST, and others. That makes it a compelling molecular handle for imaging and therapy. NeoBOMB1 basics. It's a GRPR antagonist that can be labeled with germanium-68 for PET or lutetium-177 for therapy. Antagonists often show strong tumor uptake without needing full receptor activation, useful for both detection and delivery. Preclinical work showed high affinity, favorable kinetics, and clear theranostic potential. The theranostic flow: find then treat. Find, patients first get a germanium-68 NeoB PET scan to confirm that their lesions are GRPR positive. If uptake is adequate, they can be considered for therapy. Imaging studies in GRPR-expressing tumors, including GIST, support this selection step. Treat. Therapy uses lutetium-177 NeoB, whose beta particles travel millimeters, ideal for crossfire inside tumor clusters, while sparing more distant healthy tissue. First-in-human designs focus on safety, dosimetry, and early signs of activity. Where are the trials right now? Imaging Phase 2 germanium. 68 NeoBOMB1 and GRPR overexpressing tumors. Multicenter, single-dose imaging to characterize uptake and safety. Therapy first-in-human Phase 1 IEA, lutetium-177, NeoB in advanced solid tumors. Safety, tolerability, PK, and distribution, classic dose escalation/expansion design. Breast cancer programs. PET add-on study in breast cancer to map GRPR expression in stage disease. Combination therapy trials exploring lutetium-177 NeoB plus capecitabine dose-finding optimization after CDK4/6 inhibitor failure. And a separate study combining lutetium-177 NeoB with ribociclib plus fulvestrant in ER+/HER2/GRPR plus disease. Bottom line, NeoBOMB1 isn't a single tumor moonshot. The trial map is deliberately multi-tumor because GRPR shows up across several histologies. What cancers might benefit? Prostate cancer. GRPR PET is being explored particularly in biochemical recurrence and earlier disease. Some prospective work suggests germanium-68 NeoB can outperform a comparator PSMA-2 tracer in certain settings. It doesn't replace PSMA, it can complement it. Breast cancer. Multiple trials are enrolling ER+/HER2/GRPR plus cohorts, some with combination regimens. GIST. Imaging data show germanium-68 NeoB can visualize tumor burden, even suggesting a path to therapy in TKI-resistant cases pending more evidence. Lung and others. Reviews note GRPR expression in subsets of lung and other solid tumors. Ongoing studies will define which niches are most responsive. Safety, dosimetry, and practical caveats. Kidneys. As with many peptide radioligands, renal dose is a watch item. Preclinical repeat dose work flagged kidneys as the most significant site of radiotoxicity, managed clinically with amino acid co-infusion and careful dosimetry. Physiologic uptake. Pancreas and parts of the GI tract express GRPR, expect background uptake that readers must learn to interpret. That's where experience and standardized reporting matter. General tolerability. Early human imaging data indicate good safety profiles for 68J NeoB. Therapy. Safety is being defined now in dose escalation cohorts. How does GRPR compare with PSMA and SSTR? PSMA, for example, Pluvicto PSMA PET, prostate-specific membrane antigen, dominant in mCRPC and increasingly earlier lines. Strengths: mature data, supply chain, watchouts, salivary uptake, PSMA-negative variants. SSTR, for example, Lutathera SSTR PET, somatostatin receptor, NET's mainstay. Strengths: robust survival/quality of life data. Watchouts: heterogeneous expression in high grade. GRPR NeoBOMB. 1. Receptor found across multiple tumors. Strength is breadth and a potential complement to PSMA in prostate or alternative target in breast/GIST/lung. Watchouts: physiologic GI uptake and the need to validate which patient subsets benefit most. Think of GRPR as the third pillar in Theranostics. not a replacement for PSMA or SSTR, another tool when those targets are absent or inadequate. What to watch next? Roadmap. Dose-finding readouts from the Phase 1 IA therapy study. MTD/RP2D, organ dosimetry, early response signals. Patient selection rules from imaging trials. SUV thresholds that predict benefit. Combo data in ER+/HER2 breast cancer. Capecitabine or CDK4/6 partner regimens might expand benefit. Cross-modality comparisons PSMA versus GRPR in prostate to see when GRPR wins or adds incremental lesions. Final thought, if GRPR keeps delivering, NeoBOMB1 could become a flexible theranostic that opens doors across multiple cancers. I'll keep tracking the data as it drops. Do you have any experience with this? Hit subscribe and drop your comments below.